In this video, I'm going to show you how we use our online model to explore changes in a complex system. Here, we see a simplified model of cellular respiration, and we can track the inputs, glucose, and watch how glucose's intermediates travel through the system. Glucose goes into glycolysis, which then gets split into pyruvate. Pyruvate's fate can go into pyruvate processing to be made into acetyl-CoA to enter the citric acid cycle, or pyruvate could enter into lactic acid fermentation in the absence of oxygen to make lactate. At each stage, you'll see that NAD+ is converted to NADH. So every time that happens, we are capturing high energy electrons that eventually make their way to the electron transport chain. So overall, this process should be familiar to all of us. One thing that you may notice about this chart or this image is that it contains these red lines with these stops instead of black arrows. What's going on here? So these red lines with stops indicate points at which a product such as pyruvate or an intermediate such as pyruvate can act as a feedback inhibitor to a step before it was created. So for example, here in step 4 of the citric acid cycle, NADH is produced, and NADH actually acts as a feedback inhibitor for a process earlier in the citric acid cycle. And so these red dots, or red lines I should say, are points of possible regulation due to some sort of feedback inhibition. This would be a negative feedback cycle, right, where a pyruvate slows down glycolysis until pyruvate's gone, and then glycolysis turns back on to make more pyruvate. So this would be an example of negative feedback inhibition. And we have a mixture on here of both allosteric or non-competitive inhibition as well as competitive inhibition. And if you want more details on exactly which step is which type, you can read about that in chapter 9 of your textbook. What I want to do today is I want to walk you through how to access and operate the model we're going to be using to simulate regulation of the cellular respiration pathway. Then you can do the following 3 investigations on your own using the model to simulate sort of what the response would be to a variety of scenarios, whether it's glucose starvation, fatty acid metabolism, or the absence of oxygen or anaerobic conditions. So the system that we're going to access is called learn.cellcollective.org. This is an open access resource that allows for the modeling and simulation of a variety of life science concepts. And so it's a useful simulation-based approach to learning biological processes such as cellular respiration. Founded by the National Science Foundation, so it's free to access. You as a student need to enter to create an account, add your email, your name, and a password. And for the institution, you can write San Francisco University High School, and there is a terms and conditions that apply to the use. In other words, use this for learning only. Don't publish it for any real reasons. But I'm going to go ahead and log in so that I can show you where we're working. When you first log in, you should be dropped at a bunch of public modules. These are modules that cover a lot of different concepts in biology. Regulation of cellular respiration, photosynthesis, bacterial chemotaxis, prokaryotic gene regulation. There's a lot of really useful things here. We will only be using the simulating cellular respiration one for this class, but if you ever are curious about food web dynamics, if you're curious about cell cycle regulation, these are super helpful simulations to practice and learn concepts with. But what we're going to focus on is specifically simulating the behavior of cellular respiration. And when you click on it, it should bring you to an overview, which will talk you sort of through what you're going to be looking at today, and you'll see an overview of entire process of cellular respiration, you can read some of the objectives that can be accomplished using this particular model. And we're going to go to the button that would say Start Lesson. Okay, so there is a nice little excerpt here that tells you exactly what to do, or you can go back to my lesson and see what my steps are going to be. So we've accessed our simulation, and our workspace should be set up to look just like this, which it is. So we want to provide the cell with 6 major inputs of cellular respiration, and we'll adjust the sliders on the right-hand side of the page to make those boxes click to 95%. So are any of the external components new to us? And we're going to be able to list them in our notes. So let's go back to our model. Here on the right-hand side, we have our external components, and I'm going to go ahead and click each of those components so that I can provide them in the cell. And you'll notice that as I click those components in the activity network, those pieces are given semicircles, and so they show up to become available to us in the model. But I'm going to want to switch my activity to 95% for each of them. So now I have all of my external components selected, and the activity for those components is at 95%. I'm actually going to keep fatty acid metabolism at 0 for the moment, but we will come back to use this later. Within the Activity Network panel, you can click on components to change them to gray-blue. What this does is it allows those components to be recorded in the graph, in the simulation graph. And so I'm going to click these 5 components just so I can watch cellular respiration happening in my graph. So I'm going to need to include the process of glycolysis, which makes sense. I'm going to include the process of pyruvate processing, citric acid cycle, an electron transport chain, and I'm also going to include lactic acid fermentation. So now that I've got all 5 of these processes selected, I'm going to simulate cellular respiration in my graph. I recommend keeping your simulation speed at 1 so you can watch what happens in the graph over time. And I would recommend keeping your simulation window probably around 90 to 100. They recommend 100 in this model. And then you can hit play. And what happens is this activity network mimics what would be happening in cellular respiration. And there will be some variation in the values, but once the cell or the model cell sort of achieves its overall system, we start to see leveling off of each of our processes. And so I'm going to pause right here, and I'm going to observe the relative activity level of either the 5 processes I've selected or the starting components that I've added to the network. Note here that I haven't added in fatty acid metabolism, so it remains red. But each of my starting components is bright green because they are found in high concentration. In other words, glucose should be around 95%, NAD plus is around 95%, and all of the other external components that we've added reach, the 95% that we've asked them to be. So once we've done that for all of our external components, we need to know what is the responding rate of activity for each of our 5 processes. Well, we can see pyruvate processing is fairly limited, lactic acid fermentation is activity level somewhere between sort of 40 and 60%, glycolysis activity level is always around 65%, electron transport chain and the citric acid cycle have values as well. So these are sort of activity levels relevant to one another. So keep that in mind that we don't have a real unit here on the y-axis, but it is sort of an activity level relevant to each other, or relevant to the concentration of substances within the system that we've selected for up here. And in your activity network, you can see exactly how one particular substance is used at multiple different stages, right? And you can see that there are some feedback loops that are associated with certain steps of the process of cellular respiration. And I want you to think about these feedback loops in the context of this simple diagram of feedback loops up here. Does it make sense when you look at the simple diagram that you're seeing feedback inhibition possible at multiple steps for what you're seeing overall in this activity network? When you hit stop, it will erase your diagram or your graph, and then you can allow it to run again and pause it whenever you need to. And again, to select a particular value or particular substance versus process, you just sort of hover your cursor over it. If you want to see these graphs in a little bit better detail, you can extend the graph up so that you can look at it with some more precision. So that's sort of the general way in which the model is used. Your 3 investigations are going to examine what happens when you have glucose starvation in the brain. So do a little bit of reading on how the brain uses glucose, make a prediction about what you think will be the outcome, and then test your predictions. Once you've tested your predictions, you can write up, using this as sort of a formula, the cascading effect of what you think will happen if glucose is not available to each of the subsequent processes. You'll investigate other sources of energy other than glucose. In this case, we're going to be focusing on fatty acid metabolism. So that is on the model, that is on the model here, where we can add that in and increase the amount of fatty acid metabolism that's available. And we could decrease glucose and see sort of how the model operates under those conditions. So again, you would make a prediction, test your prediction, and write up a summary of what you think is happening. And then finally, you'll do the same investigation, but now you're denying the system oxygen. So you're going to be inducing an anaerobic respiration.